Hello students, I'm Dr. Arpita De. A very warm welcome to my YouTube channel. Today we are going to discuss about one of the most important topics from developmental biology. We popularly call it programmed cell death or otherwise known as apoptosis. Mind it, the word is not pronounced as apoptosis. It is pronounced as apoptosis. The second P is silent. Okay. Now, before we come to the main point of discussion, let's first see from where did this word originate. The word apoptosis is derived from the Greek word apo, which means separation from, and tosis means falling off. This phenomenon was elaborately studied by Kerr, Wiley, and Curie in 1972. Now, apoptosis can briefly be defined as a well-regulated pathway of cell death which is induced by a tightly controlled suicide program through enzyme activations. So, it will be seen when we move on to the details of our lesson. It will be seen that apoptosis constitute cell murder or cell suicide when they lack any incoming survival signal in the form of trophic factors, or when they detect extensive damage in their own nucleus. Now, from this picture, what do you understand? This picture first show that there is an intact cell which we say a normal cell. Okay, now when will this normal cell commit suicide? A viable cell or a normal cell is there. The first thing that we get to see is the shrinkage of the cell and heavy condensation of the chromatin material. Okay, the cytoplasm become denser with densely packed organelles. Thereafter, here, what happens? We see the membranes form blebs. We call it membrane blebbing. The DNA or the nucleic acid becomes fragmented. And finally, what happens? These apoptotic bodies are generated which are ready for macrophage engulfment. Now we'll move to various types of cell death that occurs in the body of a living organism. It is not always apoptosis that is causing cell death. There are other processes too, and each of them is strikingly different from the other. Now we'll first talk about necrosis. What is necrosis now? Necrosis is an externally induced passive pathological condition that dissipates and often causes swelling. This is very much different from apoptosis. Apoptosis is something absolute different from necrosis. In necrosis, what we get to see, it is completely external. It is externally induced and it's a passive process. It's a symptom of a pathological condition that dissipates and often causes swelling. Here what happens, the cell membrane breaks and there is leakage of enzymes which causes the swelling. Now we'll come to mitotic catastrophe. It is an internally induced passive pathological condition that also dissipates and cause swelling. We have just now talked about the reason of swelling. Then we'll move to apoptosis. What is apoptosis? We had just now discussed. It's an externally or internally induced active pathological or physiological condition that neither dissipates nor cause swelling. All right. Now, when is this phenomenon taking place in your body? How will the cell know when should I commit suicide? Okay, let's see. There are two very particular reasons why apoptosis or when apoptosis take place. Okay, when cell lacks any incoming survival signal in the form of trophic factors, and second, when they detect extensive DNA damage in their own nucleus, which is beyond repair. We all know that little bit of a damage is always repaired by the inbuilt DNA repair mechanism in the cell. There is proofreading, checking, all these phenomena that operate in the cell, which checks the error and rectify them or make the DNA damage free. But when the cell realizes that the damage is beyond the capacity of their repair mechanism, that is the point of time when they plan to commit suicide. All right, now we'll go to the conditions that lead to apoptosis. Fine. What are the conditions now? The conditions are of two types. One is physiological condition, the other is pathological condition. Okay, we'll first talk about physiological condition. First is embryogenesis. Now, what special role do you think this phenomenon plays in embryogenesis? In embryogenesis, it helps in the formation of proper independent digits. I think you must have seen people having clustered digits. That means fingers are joined with one another. In those people, the mechanism of apoptosis did not operate. That is the reason probably their fingers got joined to each other. Okay, so unnecessary cells that were there in between the digits were all killed by this phenomenon of apoptosis. Then we'll come to involution of hormones. This happens in the menstrual cycle of female. There is atrophy of ovarian follicles at menopause. This is also caused by apoptosis. Now, loss of cell in proliferating population. Okay, bone marrow thymus constantly divide, so constant number is to be maintained in order to maintain the hemostasis. Okay, the bone marrow in our body and the thymus gland is constantly producing cell, and if they go on producing cell, there will be an exponential rise in the population of cell in our body, which is not expected. Therefore, a certain percentage of cell should be driven out of the system as well. This is carried out by apoptosis. Next, we'll come to removal of self-reactive lymphocyte. If self-reactive lymphocytes are there, what will happen? They will create an autoimmunity in our body, right? So these few points are taken care of by apoptosis. Now we'll come to the pathological condition. What happens here that leads to apoptosis? First point is DNA damage. DNA damage means what? We were just now discussing that when there is extensive DNA damage which is beyond the capacity of repair, at that point of time, the cell decides to switch over to apoptosis or suicide. Fine. Now what is next? Misfolded protein accumulation. Protein folding is one of the most important factor that controls normal growth and development. When there is a misfolded protein, at that time a cell should die. Otherwise, this will lead to various diseased conditions. Then comes viral infection. There is nothing much to explain. And then comes duct obstruction. When there is an obstruction in the duct, so this obstruction can be removed by what? It can be removed by removing that obstructive particle, by removing that obstruction from the duct. Who is removing this obstruction? This obstruction is removed by apoptosis. Fine. Now we'll come to these pictures. Let me mention the source of these pictures are Google. Okay, these pictures depict certain phenomena that occur in embryogenesis of certain amphibians, that is tadpole, and in human as well. Here, what happens, you see, I think you all have seen that a tadpole has got a tail, but an adult frog doesn't have that. Why is this happening, you think? The tadpole in its young stage can swim in water, so it needs the tail. But as it becomes an adult, there is resorption of tail. The tail is no longer needed and the cell over there dies out by virtue of apoptosis, and this adult frog emerges without a tail. Now we'll come to this Mullerian structure. Mullerian structure is absent in male and present in female. It is found in female fetus. Sertoli cells in male fetus secrete a factor named anti-Mullerian factor that inhibits or prevents the formation of Mullerian structure in male. This is also happening by virtue of apoptosis. Here see organ sculpting. We had just now talked about that. See, in between these are about to form digits. These bulges will form the digits. Okay. Now, from this diagram, what we see that it's just looking like a webbed chunk of tissue. These violet dotted parts are places or locations where apoptosis will take place. See, these valleys are cleared off the cells and ultimately free digits are seen. Here, look at this part. Look at this part of tissue. Here, apoptosis is not complete. At the anterior part, it has taken place, but unfortunately here in the posterior part, it did not take place. As a result, the digit did not become free. Okay. Now look at this, self-reactive cells. These self-reactive or self-antigen recognizing cells are removed from the population by apoptosis. All right. So thank you so much for your kind attention. This video lecture is a bit long. That is why I thought of dividing apoptosis into two parts. This was the first part of my lecture, and the second part will be there, which I'll be uploading soon. Thank you so much.